Yeah, welcome everybody for our fourth annual Invicro software user meeting. Thanks for coming here, and also everybody that joined us online for today's session. We already had a great workshop around VivoQuant scripting yesterday, which many of you attended. That I heard was very successful and was well received. And today we wanted to present to you a little bit of our work that we have done over the last year, as well as give you an outlook on what we as the software group at Invicro are focusing on, and give a couple of our users also a chance to present on their work, which very likely have similar problems that you have and used Invicro solutions to solve them. And I also hope that we have a lively discussion about things. So please ask questions. This is also more like a workshop where we can also go into more details of things that are of interest to you. So please feel free to come up with questions. We are also, that's not working yet. Yeah, we are also celebrating actually today the 10-year anniversary of Invicro. To the day, 10 years ago was our incorporation of the Invicro LLC, and we are having some internal celebrations also. But we also wanted to share this with you, and we should have a drink on Invicro later at the bar downstairs. I hope you brought the champagne or something.
Champagne if we want it.
So, and with this, I would like to pass on to Bill for the introductory talk. Thank you.
Thanks, Krishan. And thanks everyone for joining today, both in person and online. We are excited for a robust discussion around, as Krishan said, how users both internally at Invicro and externally at pharma and academic institutions are collaborating with each other to solve these challenging data informatics problems in an effort to answer these even more challenging biologic questions and advancement of their drug development programs. As a play on our 10-year anniversary, I thought it'd be interesting just to look at how we got here. Our marketing group did a nice job kind of recapturing a brief timeline history of Invicro, and we've gone through a lot of change, or especially over the last couple of years. From acquiring companies, merging with groups, specifically around the scientific and operational capabilities of all these teams. And, most recently with the acquisition by Invicro by KM, really wanted to just highlight on a couple comments on what that means moving forward for our users and all the scientists that we work with day to day. Konica, after acquiring both Ambry Genetics and Invicro, really set out to form a advanced precision medicine group that is combining world expertise across different bioinformatics, quantitative biomarker disciplines, from genomics, histopathology to radiology. And Invicro, as we look at how we support and fit into that platform, we really view ourselves as a key data analytics technology driver of how to best leverage the current practices in managing data, viewing and analyzing data, reporting on that data from a variety of different modality types, whether it be a variety of modalities within radiology. With Konica Minolta, we've been advancing into the histopathology space more, and how best we can collaborate with our Ambry colleagues, as well as, I think, the quantitative biomarkers at pharma that are producing other data sets that groups are wishing to merge and to analyze in a cross-modality setting to answer these complex questions. And so the tools and the people that we have organized across the enterprise will continue to form into a group that will be made available to you as customers from a deployed software standpoint, as well as the continued engagement we have with many of our customers in the pharma data analysis space for the contract research services we offer as well. So very exciting times here at Invicro, and we continue to make investments in our company to stay at the cutting edge of how we think about data handling, data processing, and general data analytics practices. A little more specifics on Invicro. We continue to stay as an organization laser focused on radiology and increasingly some histopathology-based assays and the data handling tasks, data processing tasks that come with that. What may have gotten hidden from the large acquisition from KM was us joining forces with the Imanova team and their scientific group and operational capabilities, their site capabilities, Core Clinical, who their team has a wide breadth of experience in late stage imaging core lab services, particularly on oncology, but other therapeutic areas. And so the challenges that our software team and the image analysis group here at Invicro kind of jointly face to help support the wide array of different types of data, again, from a data handling, data processing, data analytics standpoint, are only getting more complex. And we're looking forward to exploring how best to provide solutions, not just internally, but working with you as well, as we certainly appreciate and enjoy all the scientific collaboration with you as customers as we try to jointly build out these solutions to meet each other's needs. And how we envision the future building and supporting the wide array of these types of data sets is really continuing focus on the core software platforms that we develop in-house. But I think you'll hear a great theme today of how our software technologies can be incorporated into a wider picture, a larger footprint or ecosystem of different software tools for handling these different types of data sets that may fall within the radiology, histopathology category or complement to such. So you may be familiar with this diagram of how, IPACs currently and VivoQuant as a platform handle a variety of different preclinical and clinical data workflows, whether it's from a data sharing standpoint, data transfer standpoint, or from the nuts and bolts of viewing and analyzing the data. We continue to see IPACs as a core platform to manage and be a content management system for all things data related in imaging trial. And how we see the roadmap building out is where we can make investments on improving integrations to not just our own software, but other tools that groups are using so that IPACs continues to be a center hub of the wheel, so to speak, of how you think about managing your image data. With that being said, the current environment is very interesting in how the landscape is evolving of how groups are thinking about managing not just image data, but all types of data within pharma. And as you can maybe see from a few slides ago, even at Invicro and working with our Ambry colleagues and our KM colleagues of how can we best think of IPACs as a component of a larger data management strategy so that groups, when you have this single question of, can I look at a correlation of data output from a variety of quantitative biomarkers, that the technology and the infrastructure is not limiting to how you're accessing that data, how you're interacting with that data, how you're analyzing that data. And so that is really a core focus, and I think will be a theme of the year in how we work with groups, our current customers, our hopeful new customers we don't know yet, but hope to meet soon and even solve challenges internally. And I think what continues to be such an exciting time at Invicro is the symbiotic nature between the problems that are presented to us through our contract research business and the problems that we interact with our customers using our deployed versions of software. We really see that line getting more gray in how we think about supporting these data handling, data processing, data analytic challenges. And so with that, I think, it's a great way to set the tone for many of the themes you'll hear in these talks today. I'll try to shut up quickly because we have a great lineup of speakers that will be touching on a variety of these topics. But, I think it's important, especially at a day like today, to really make sure that from Invicro, you realize how much we thank our relationships and our collaborative time with you as our users, and also our own team. So there's a lot of people here today, hopefully, that users, customers get a chance to meet. You see myself, TJ, Rhonda, Melissa as really been the main interface to you in a lot of these projects, but there's a huge team behind us that helps support a lot of the product development, the features development, general application support, technical support. And so it really, I hope everyone gets an opportunity to interact with the entire team here. That's from our side. And then again, we really should be thanking our users. You help drive our success. So with that, a little play on one of the first marketing campaigns we ever had at the company. If anyone caught it, I think it was back in 2013, but it was highlighted by a phrase, "See more." So let's help each other see more today, one voxel at a time. So with that, I'd like to introduce our first speaker, Dr. Jacob Hesterman, current head of research and development at Invicro. And he'll be presenting on VQ applications within the core lab and the challenges that his team faces in supporting these data problems. Let me just give you a slide. This mouse is...
Yeah, okay, great. Thanks, Bill. Thanks, everybody. Okay, so actually I'm going to start with some acknowledgements because, what I'm about to show is the work of a huge team, software core lab, but I especially wanted to point out Dr. Tyler Wellman. He should really be presenting these slides. He just couldn't be here today. A lot of what you're about to see has really been, these are sort of how we're supporting applications using the software tools that have been made available to us by the software team. I'm going to focus particularly on VivoQuant in the core lab. Today I'll mention a couple of other things, but that's the real focus. So in terms of a brief outline, I want to give a little bit of an introduction to our core lab. I think most folks are familiar, but just very briefly, and then talk in particular about a few, really it's going to be very case study heavy. We're going to hit some examples, basically how we're supporting sites and data management with VivoQuant, a couple quantitative analysis examples, and then a couple clinical read examples. Okay, so core lab, just very high level, obviously, with these little cartoons, but essentially, and we're going to drill down a little deeper. If we have typically a pharmaceutical sponsor, ourselves as an imaging CRO and some clinical CRO, we're going to be interacting to meet some sort of multi-center imaging trial endpoint, often late phase, some early to mid phase, interacting with multiple imaging centers who are going to then provide data in whatever format to the CRO and ourselves. We'll then provide it back either to the CRO or to the pharmaceutical sponsor, and eventually to the FDA, which is a critical component. Any questions so far? Actually, while we wait, will people who are listening online be posting questions that'll get asked?
Yes.
Yeah. Okay.
We'll read those out.
So I should leave a few minutes, right?
Yeah. At the symposium, I had to stop sharing and reshare a few times. Something with Zoom.
Weird.
Yeah. But I'll note that we do that.
I'm just going to go to the next slide so I can speak to it for a little bit.
Okay, there we go. We're all set.
It's working?
Yeah, thank you.
How do I? Okay, great. So just drilling down a little more deeply, and this is really what we're going to be, I guess I should stay here by the microphone. This is what we're going to be focusing on. So data coming into us, we need to get that data. We typically need to do some sort of image QC standardization steps, get that data stored, perform a read, some sort of quantitation, maybe both, get that output back on a repository and get it out. So I'm really going to focus, I think you're going to hear a lot, especially from Christian, for example, on these two areas. I'm going to be focusing on standardization and image QC and reads and quantitation. The IPACS is really the foundation we're building a lot of this on. We use multiple softwares with the IPACS in this way. Today, I'll be focusing on the VivoQuant. a few things to keep in mind as we look at these examples. So the primary requirements are, we want to have accurate tools. And here I'm really talking about quantitation. I mean, reads too, but we want to have accurate output. We need to be robust. We're going to be getting data from lots of centers, different pathology at work. You know, we need robust tools. And then we need these tools to be compliant. And really they're what we want. We want a nice audit trail. Some other things that are nice to have, but that really means we actually have to have them as well. These have to be efficient. We're going to be churning through it. Last year in our core lab in New Haven, the team processed over 10,000 amyloid PET scans alone. So we need efficient tools. We're going through a lot of data. We want to, errors are going to happen, but we want to put in place processes that will at least try to limit the opportunities for users to make errors. We want to track how we're doing and where data are. We want these tools to be user-friendly, and then we want to be able to extend. You know, we're trying to, I think as Bill made clear, we're moving into a lot of different areas, and we want to be able to support a lot of those studies just generally in the core lab, but also within VivoQuant in the core lab. So the first couple examples are going to be on that really site setup, and then I have some acronyms here, TQC and SQC, technical quality control and scientific quality control. This is all, it comes down back to that sort of idea of garbage in, garbage out. We do a lot of work up front before we even get to the point of trying to analyze or read these data to try to make sure we have good data going into the system. And we're using VivoQuant in all of these steps. So I'll start with examples of phantom data. So multi-center imaging trials, often with a quantitative component. We spend a lot of time trying to make sure that, one, the imaging centers are set up properly to acquire and reconstruct the data that we need. Actually, again, we've set up over, this is really the work of Justin Albani and his team, we've set up over 900 sites worldwide for acquiring this type of data. We then want to pull in that data, and given us, often we'll have what's referred to as a study-specific target spatial resolution, let's say. So we want to get all this data in from sites, characterize that data, and then figure out what we need to manipulate in terms of each data set coming in from those sites, such that they'll be harmonized on the back end before we do any analysis or reads. So what we've built are custom VivoScript pipelines, such that as these phantom data come in, we're storing a lot of the site and scanner information on the IPACs and pushing the data, JZAC phantoms shown here, but also, for example, Hoffman phantoms through automated VivoScript pipelines that extract all kinds of both qualitative images of the data, but also all kinds of quantitative data to support this harmonization effort. How is this practically useful? So I'll give an example. It's actually one of my favorites right now. So we're working with a group, and they're running a couple studies. It's going to have probably 250 to 300 sites worldwide acquiring data. And there was discussion of, should we go physically visit all of these sites to set them back up? That's a huge amount of effort and energy, time, expense, et cetera. And you know, I feel all things in moderation. It's probably useful to set up some of those sites, but to set up all of them again physically would be a lot. So what we were able to do is, on a rolling basis, as sites are enrolled in the study, we get the new sites every 2 weeks that have been enrolled in the trial. We compare those sites against the sites we have in our database, automatically extract the phantom data from those sites, push them through this quantitation pipeline. And actually what you see here is a page from an example PDF output that's automatically generated that includes an image of the phantom, quantitative summary statistics for that phantom, and then based on some predefined criteria that we arrived at with our collaborator, if that site was approved, should be discussed, or needs an on-site setup. And by utilizing this pipeline, we can provide an updated PDF every 2 weeks, and we've been able to set up 80% of these sites remotely and take that number, you know, down to 20% on-site visits. So this has had a real practical impact on day-to-day workflow. I'm just going to give, so terminology, technical quality control. Basically, we set up these sites. Data are now, we've done the phantoms. Everything looks good. Data are now coming in. Before we process or read, technical quality control, basically looking at the data, trying to make sure there's no missing slices. We were told we got an MR of the head. Is it really the head? You know, sort of basic, like, the site should have used these acquisition and reconstruction characteristics. Have they done it? That level of quality control. When that passes, it goes to a scientific quality control, which is more of a domain expert who's looking at it and assessing, is this scan suitable for the purpose, for purpose, from a quality standpoint or things like that. I'm just going to walk through one example of a technical quality control. So this is MR data. So what you can see here is actually, this is just a little zoom in on the VivoScript bar. This is all, a lot of these VivoScript. So settle in, you're going to see a lot of images of forms. I hope the screen is big enough. But here, MR technical quality control, this very small thing says, please select the subject to run, loads data associated with that subject. And then this is a form, and you're going to hear a lot more about the underpinnings of this, how this works later on. But essentially, this is the form that the user would see within the VivoQuant, but is actually on the IPACs. It's a mixture of auto-populated. So these grayed out fields are auto-populated. And then the user can go through and fill in the rest of the form for storage on the IPACs. And one of the features that's really nice, a couple things. If we come back to those criteria for what we want in our tools, we need them to be easy to use and efficient. So here, for example, it's very nice. This, I realize how small this must be. This just says basically, are there resting state fMRI? If the answer is no, that's the only question. If you click yes, then the form expands and you can enter in the details specific to that acquisition. Similarly, kind of maybe complementary, if you neglect to fill in a field that you're supposed to and try to submit, the form will come back and say, whoa, you're not done yet. So this has been a really useful tool. These are screenshots that are just showing, again, we're doing a lot with workflows, which I think will come up again later as well. So essentially, once we do successfully submit this technical QC, I mentioned that there are additional steps. In this particular example, the next step is a read for ARIA, which we'll show an example of later. You can select which user is going to be performing that read. In this case, Yannick or Rhonda, two clearly key people who could do this ARIA read. And then once they're selected, actually there's a Slack notification to the user on our team that says, hey, you've been assigned this task, go get after it. Okay, so now we feel pretty comfortable. We've gotten the data from the sites. We've made sure that it's okay for what we're trying to do. And now let's do some analysis. And here is where, I wanted to give a couple examples where, we're lucky to have really strong collaborators so that we can both perform this kind of work to meet the study aim, but also often work with our collaborators to do some research and development tasks. So this is an example where we've been, as a complement to criteria-driven reads, we've been doing a lot more quantitative analysis of oncology data in the core lab. What we typically use as a starting point is PERSIST, which is PET response criteria in solid tumors. It's familiar, it's a good base, and we can build upon that. So actually, these examples are from a multiple myeloma study, and if people were here last year, they would have seen some of the beginnings of this work where we adapted PERSIST to support this analysis. So it's analogous to RECIST, typically that's used in CT and MR anatomical assessment. It's been around a number of years. But what we wanted to do here was that, okay, PERSIST, we're going to look at SUL peak, so SUV lean body mass, the peak values, how they change over time. This is these are really the endpoint, maybe some other metrics. But it still leaves the question of how are we going to define the lesions from which we're going to extract these quantitative values. And here what we're doing is actually looking at a couple approaches, both of which are totally driven by VivoScripts. One is a little more objective and rule-based, where we're saying basically we're going to flag all the lesions automatically identified with an SUL peak value above some threshold and a value within some range. And I'll talk more about that. The other is a little more subjective and user-driven, where the user is scanning through the image and looking for the lesions and segmenting them. So this schematic, basically, I won't go in gory detail here, but essentially it's an automated iterative pipeline that the user actually clicks one time in a reference region to define a threshold. And then the tool essentially loops through and says, for as long as I find a voxel above a particular threshold, I'm going to generate an isocontour around it. If that resulting contour is within an acceptable volume range, that's going to become a candidate ROI of a lesion. If not, it's going to be rejected, and I'm going to repeat this until there are no voxels left. Basically, the result is a set of regions of interest, some of which are obviously not lesions, and some of which probably are. This tool is tailored specifically to be overly sensitive because we wanted to avoid false negatives because that's problematic downstream. And then a radiologist goes through those lesions one at a time and says, I'm going to accept that as a candidate lesion, or I'm going to reject it with some rationale. So that later, if someone comes and looks and says, why didn't you keep this lesion? There's a rationale, and that rationale won't be because it didn't give me the answer I wanted. Now, the concern there was it's highly objective. It's very quantitative in nature, but it might not be sensitive enough. We're actually not sure. So we're going to process in a couple studies data the same way, but now allowing a user within a VivoScript to go through, find a lesion, click on it. Again, isocontour-based segmentation with manual editing if needed to accept lesions that way. And the way this looks is you essentially see a user. I'm glad I put these here because you can probably actually read those. It's literally click, doesn't have to be in the hottest part of the lesion, just has to be nearby. You get a auto segmentation based on some threshold. You can easily adjust that threshold to a predefined value or a custom value, and then manually edit that ROI or just directly save it. For both of these pipelines, all of that information is stored back up onto the IPACs in the form of data points. So, we think this is neat. At the end of the day, we're going to have two very distinct pipelines that we can use in our validated core lab for segmentation of these lesions as quantitative accompaniments to any kind of read. And then we're also going to have comparisons between those measures in a couple of studies, both to one another and to some clinical outcomes. So we think this could be a really informative way to develop optimal pipelines for future studies. I'm going to keep going on in the interest of time here. Another example, something we do a lot of, and I don't think it's typically thought of as a core lab study, is dosimetry. So we do a lot of radiation dosimetry at Invicro because we do so many first-in-human studies. And last year we put 12 radioactive compounds in humans for the first time, 6 in London, 6 out of the US location. So we do this a lot, and dosimetry is a part of that. There are cases where we need to run later phase dosimetry studies, often for safety. And we've built VivoQuant pipelines to do that, which is really exciting. Now here, the most time-consuming step is actually this region of interest generation. I'm not talking about this aspect of the work today because it's more on the development side, but we do actually have extended our automated tools to support MRI and CT whole organ segmentation in support of these kind of outcomes. But basically, what we have now are pipelines where regions of interest are generated, and then the VivoQuant picks up the data in the regions and actually executes all of this back end automatically. So from a visualization standpoint, there's not too much to say because essentially VivoQuant is running in the background as a VivoScript. It's picking up regions, data, computing mean residence times, generating input files that are Olinda ready, pushing them to Olinda, pulling that data back out, and storing everything back onto the IPACs in the form of data points. And this was actually the point that I really wanted to drive home, is that as part of it comes back again to those bullets we showed at the beginning. Here, one of the tricks is we want something that has a nice audit trail that can really be traced. And what we have here are automatically generated data points. And I know these are probably very hard to read, but we have input data. You see this is a QC file. This is an output file, input file. All of the essentially measures, metrics, regions, inputs and outputs are stored in the form of data points. They have that user stamp. They have the version stamps. We're able to report on them efficiently. So this gives us a nice, robust workflow for this kind of task. Okay, clinical reads. So here, I'll mention, I do want to mention a couple of the other tools, and you'll see how we're using the IPACs with some of these other tools a little later. I think Rhonda's going to touch on that in particular. We're using the VivoQuant more and more. We use PMOD a lot internally still with our neuro studies for analysis. Also for criteria-driven reads in particular, we're using MINT Lesion, and it's communicating really well with the IPACs. You know, those criteria-driven reads, we don't necessarily see the need to reinvent the wheel. And, you know, that's a good package that we can use. But we often have studies where either a couple things. One, if we're doing a quantitative analysis, from an ease of processing, it can make sense to perform a read just within the same software. And two, we often get kind of off-the-wall requests, or there are very complex read paradigms. And because we can have full control over the VivoQuant development, we're able to support those requests very nicely. Now, that said, the focus of the VivoQuant is still really for research analytics, and that's our primary driver. Clinicians don't necessarily feel that way all the time, so we do have to have many, many options for them to view data. They want to see things a certain way. This is just a very quick slide to show if people are using VivoQuant and they haven't played with multi-view recently, it's pretty awesome now, and you can really look at the data in any way, shape, or form that you want, and people ask for it. This slide is just another form, again, saying nothing fancy here other than a nice case report form. That this is from the same example earlier, and this was a case where it just made a lot of sense. If we're doing the quantitative analysis and we have the data loaded, let's pair that with the qualitative read, and we're able to do that without any issue. This is another example of form sophistication that I wanted to just briefly touch on. So this is actually cascading directly down from that technical QC example that we saw, where there's an ARIA assessment happening. Again, you can see I've grayed out some of these fields, but you can see that several fields are auto-populated, and these were just meant to be, again, examples of how this form can work. So you can see almost the entirety of the whole form here, 6 questions. If they're all no, you're practically done. If there's a yes, then the form expands to allow you to provide more granular information about the situation at hand. Yes, no. If you hit yes here, then you can name these are anatomical locations where, in this case, ARIA-E was observed. If we're talking about age-related white matter changes, and we hit yes, here you have an even nicer situation where you have drop-down menus for the left and right hemispheres, different brain regions. You select the severity or the class of the ARIA that's observed, and then you get an automated age-related white matter score, change score, computed here, shown to the user, but grayed out and not editable. So we have a form with either essentially the amount of granularity that you need, which is very nice from a usability standpoint, and then with it, at the same time, you can apply labels and annotations to all these different regions, select from a drop-down menu the characteristic associated with those annotations, and store those all on the IPACs. So within one workflow, we have the annotations, all of the form data, everything posted back to the IPACs. So, you know, this is a bit of a cheesy conclusion slide, but basically, you know, I wanted to show here is really where we're able to, you know, we had those generic icons before. Practically, we're able to use the VivoQuant, you know, for these items in a number of ways. And just one little teaser that I think people are familiar with. This is something that, my colleague Roger Gunn, he and I co-run the analysis team here at Invicro. He and Alex Whittington have developed this very elegant and powerful tool called Amyloid IQ for analyzing amyloid data. I think some folks have seen this. It's a domain knowledge-based algorithm to quantify amyloid load. Works in florbetapir, florbetaben, and flutometamol. Very powerful, both cross-sectionally and longitudinally, and completely automated, so it could be used in supportive reads. If you look, these are just examples of kind of a case where you have a low A-beta signal, and you see about 15% amyloid load, moderate around 44%, high around 65%. And if you look at those images, they look, you know, they came out of that custom pipeline, but if we kind of just look at those, I think you could see pretty easily that they could have come right from VQ. And so this is coming soon, Amyloid IQ meeting VQ. So keep an eye out for that in the core lab. I'll take any questions. Thanks. Yeah.
So I have just 2 questions. With the one that you showed,
Yeah.
during the process of, you know, depending what was selected, are those forms still out or they fill out, internally online?
Yeah, great question. So I didn't cover the submission IPACs, which you'll see a little later. That's where when sites upload the data, there's a form that they fill out, a web-based form on the submission IPACs that contains some information. The forms that were shown here are filled out internally by our image quality control team.
And then another
Yeah,
question
please.
was, in regard, I know you mentioned that you're working with a lot of collaborators throughout, especially like the first piece of this. Are there other companies that are utilizing this technically and not going through Invicro or lab?
So, and I realize I should probably repeat the question for people who are online. So the question was, we're working with a lot of collaborators. Are any of those collaborators using these tools themselves in support of Core Lab, or for the most, or are we just using them ourselves internally? The VivoQuant and the IPACs are used broadly in terms of for supporting imaging studies. In terms of as part of a Core Lab, that's something that's currently really owned by and used by Invicro. Yeah. Any other questions, comments? Feel free to catch up with me in any of the breaks or anything, and I'm happy to answer any questions then. Thanks.